Good morning all. So today we are going to discuss about cellular respiration in bacteria. In the last session we have discussed about the catabolic and anabolic reactions that are coming under the introductory part of bacterial metabolism. Inside every cell of all living things, energy is needed to carry out every life processes. Energy is required to break down and build up molecules and also to transport many molecules across the plasma membrane. All of the life's work needs energy. A lot of energy is also simply lost to the environment as heat. The story of a life is a story of energy flow. Its capture, its change of form, its use for work, and its loss as heat. Energy, unlike matter, cannot be recycled, so organisms require a constant input of energy. Life runs on chemical energy. Where do living organisms get this chemical energy? The chemical energy that organisms need comes from food. Food consists of organic molecules that store energy in their chemical bonds. So the energy is stored in the chemical bonds within the food. Glucose is a simple carbohydrate with the chemical formula C6H12O6. It stores chemical energy in a concentrated, stable form. In your body, glucose is the form of energy that is carried in your blood and taken up by each of your trillions of cells. Cells do cellular respiration to extract energy from the bonds of glucose and other food molecules. So cellular respiration is undertaken by all the cells to extract energy from the glucose and other food molecules. Cells can store the extracted energy in the form of ATP, which is called adenosine triphosphate. Now, what is ATP? Its structure is more complex. A in ATP refers to the majority of the molecule, adenosine. It is a combination of a nitrogenous base and a 5-carbon sugar. Then, T and P indicates 3 phosphates linked by the bonds, which hold the energy actually used by the cells. Usually only the outermost bond breaks to release or spend energy for cellular work. An ATP molecule shown in the figure, it is like a rechargeable battery. Its energy can be used by the cell when it breaks apart into ADP and phosphate. So the energy which can be used from the ATP when it is broken up. And then the worn out battery, ADP can be recharged using new energy to attach a new phosphate, and again it can be rebuilt to ATP. So it says it is like a rechargeable battery. The materials are recyclable, but recall that energy is not. ADP can further reduce to AMP. Then that means adenosine monophosphate and phosphate, releasing additional energy. As with ADP recharged to ATP, AMP can be recharged to ADP. How much energy does it cost to do your body's work? A single cell uses about 10 million ATP molecules per second and recycles all of its ATP molecules about every 20 to 30 seconds. This is the structure of ATP. It possesses alpha phosphate group, beta phosphate group and gamma phosphate group, which is attached to a sugar molecule, which is a pentose sugar known as ribose. Now, what is cellular respiration? A cellular respiration, it is the process by which living cells break down glucose molecules and release energy. The process is similar to burning, although it doesn't produce light or intense heat as a campfire does. This is because cellular respiration releases the energy in glucose slowly in many steps. It is not a fast reaction. It uses the energy that is released to form the molecules of ATP. So the energy is released to form the molecules of ATP, the energy carrying molecules that cells use to power biochemical process. So the ATP is the energy carrying molecule which takes out the biochemical processes. Cellular respiration involves many chemical reactions, but they can all be summed up with this chemical equation. C6H12O6 plus 6O2, which gives, that is, glucose plus oxygen gives 6 molecules of carbon dioxide plus 6 molecules of water, and energy is released. Where the energy that is released is in the chemical energy, that is in the form of ATP, the thermal energy as heat. The equation above shows that glucose and oxygen react to form carbon dioxide and water, and also the energy is released. Because oxygen is required for the cellular respiration, it is an aerobic process. So the cellular respiration, it occurs in the cells of all living things, both autotrophs and heterotrophs. All of them burn glucose to form ATP. The reactions of cellular respiration can be grouped into 3 stages. First one, glycolysis. Second one, Krebs cycle or citric acid cycle. Third one, electron transport chain. So first is the glycolysis, this portion. Then second one, the transformation of the end product of the glycolysis, that is pyruvate to acetyl-CoA. And this acetyl-CoA enters the Krebs cycle. Then the Krebs cycle, the intermediates are used in the electron transport chain. Cellular respiration takes place in stages. The process begins with a molecule of glucose, which has 6 carbon atoms. So, what happens to each of these atoms of carbon? Let's see. Glycolysis. The first stage of cellular respiration is glycolysis. It usually takes place in the cytosol of the cytoplasm. The glycolysis is the metabolic pathway that converts glucose, that means C6H12O6, into pyruvate, CH3, COO, COO-, which is pyruvic acid or pyruvate, and a hydrogen ion. The free energy released during this process, it is used to form the high energy molecule ATP, which means adenosine triphosphate, and NADH, which is a reduced nicotinamide adenine dinucleotide. Glycolysis, it is a sequence, often enzyme catalyzed reaction. Most monosaccharides such as fructose and galactose can be converted to one of these intermediates. The intermediates, so the glycolysis is the process in which most of the monosaccharides such as glucose and fructose can also be converted. The intermediates may also be directly useful rather than just utilized as steps in the overall reaction. For example, the intermediates in the glycolysis, example, dihydroxyacetone phosphate, which is also known as DHAP. It is the source of glycerol which combines with fatty acids to form fat. And it is an oxygen-independent metabolic pathway, glycolysis. The wide occurrence of glycolysis indicates that it is an ancient metabolic pathway. Indeed, the reactions that constitute glycolysis and its parallel pathway, which is pentose phosphate pathway, occur metal catalyzed under the oxygen-free conditions of Archean oceans and also in the absence of enzymes. In most organisms, glycolysis occurs in the cytosol. The most common type of glycolysis is Embden-Meyerhof-Parnas pathway, which was discovered by Gustav Embden, Otto Meyerhof, and Jacob Carol Parnas. So the name Embden-Meyerhof-Parnas pathway. The glycolysis pathway, it can be separated to two phases. First one is preparatory phase, where ATP is consumed. Second phase is payoff phase, where ATP is produced. In the first stage, the ATP consumption takes place. Second phase, ATP production takes place. First is about preparatory phase. The preparatory phase, the first 5 steps of glycolysis are regarded as preparatory or investment phase because they consume energy to convert the glucose into 2,3 carbon sugar phosphates. The first step is phosphorylation of glucose into glucose 6-phosphate by the family of enzymes called hexokinase. These are the enzymes responsible for converting glucose to glucose 6-phosphate. In this reaction, ATP is consumed, but it acts to keep the glucose concentration low, which promotes continuous transport of glucose into the cells through the plasma membrane transporters. In addition, it blocks the glucose from leaking out. The cell lacks transporters for glucose 6-phosphate and free diffusion out of the cell. It is prevented due to charged nature of glucose 6-phosphate. Glucose may alternatively be formed from the phosphorylasis or hydrolysis of intracellular starch or glycogen. In animals, an enzyme, isoenzyme of hexokinase called glucokinase is also used in the liver, which has low affinity for glucose. That means Km value in the vicinity of normal glycemia and differs in regulatory properties. Because of the different substrate affinity and alternate regulation of this enzyme, it acts as a reflection of role of the liver in maintaining blood sugar levels. So first step, glucose converted to glucose 6-phosphate with the help of enzymes hexokinase or glucokinase. Both of them are transferase. Glucose 6-phosphate is then rearranged into fructose 6-phosphate by glucose phosphate isomerase, and fructose can also enter the glycolytic pathway by phosphorylation. So this is all about. So we are stopped the discussion up to it. Next part we can discuss in the another class.